that why block the drug with 100% inhibition? Why not you take 90% inhibition? You have got some teaching material. You have seen that graph at various concentration. So you can work at 1/10 or 100th concentration and inhibit. But you block 3 places. Okay? And then you should be able to get back and inhibit. But the problem is, if you do that, so if I now use the same picture and say I want to design a drug which will bind to all 3 targets. Now they are not off targets. It's a 3-dimensional molecule. It looks like this. This surface will go and bind to one protein, this will bind to another, and this will bind to another. And all 3 are essential. Therefore, there will be no surface left for it to bind anywhere else. Clear? So no side effect. And therefore, can I create this fused molecule? That is my hypothetical fused molecule, which will bind to 3. And if these 3 are an important pathway, then fantastic. That's right. So they will all block the whole thing. So you just think about it. So the molecule is small. Now you can see, you still can take this portion to bind something, this portion to bind something. If you really go back and see the crystal structure of aspirin, you will discover it binds to 7 sites. It doesn't bind to one site. So if your off target becomes target, then you bring the population to them. Okay. So therefore, can an inhibitor be designed? And I gave this job, and I'm very happy to say there are some people who picks it up and start thinking and figuring out a Shikimate pathway. There are proteins which can be you can take pieces and connect. So this is an important challenge. So what I try to give you an example, there's nothing magical chemistry here. Chemistry is very simple. Chemistry is nothing but charge and shape complementarity. As long as charge and shape is complementary, it will interact. Protein doesn't know it is protein. Nucleic acid doesn't know it is nucleic acid. You think it is spoon. Right? But if I don't tell you it is spoon, you have never seen a spoon. And I tell you it is a screwdriver. It's a can opener. You'll agree, no? Right? And I can show you a new use of it. I can take a stone. You have not thought about that. And now I want to puncture. So you are thinking that this has been designed and manufactured for this purpose. But yourself is using it for many purposes. You are using it to open the can, tighten a screw, create a hole. When your cutter is not working, you puncture, no? Ghee dabba. So oil dabba truck se puncture kar diya. So therefore, nature also has evolved. So your jaundiced view of one perception will not work. So chemistry of life is not so complicated. It will be surprised. The chemistry actually, if you look at the periodic table, how many elements of that are used in biology? How many elements? Hydrogen, carbon, oxygen, nitrogen, sulfur, phosphorus, khatam ho gaya. Very rarely one arsenic, and they're very rare. Life is nothing but chemistry of 6 elements. पूरा chemistry पढ़ने का जरूरत नहीं है. No need to study the whole chemistry. Mr. Jalil may go on telling study chemistry. You say I study only 6 element chemistry. Correct, Dr. Jalil? So therefore, life is very simple. And what are you learning? You are actually looking at the same small molecule which is going into this cavity and sitting there. And that's all you are doing. You are only, the problem is at this moment you are doing one at a time and creating confusion. But once you learn how to do one at a time, but you have to do then three at a time. Correct? So you have to put it, then bring other protein to it, take another protein to it and see how it works. And that's the ultimate target. And that is the real challenge. So you are actually building up steps of building the temple. So these are examples. I'm just giving you, all of you have seen these examples. So therefore, if we can move on from tuberculosis to keep low-cost drugs in the affordable by figuring out what he was telling, using chemical genomics to figure out which one will have a side effect for whom and which will not. But that's a secondary phase. We'll not discuss it today. I'll answer you with the question answer.